forums. Oh, I see. Well, welcome to Evening with the Genome Center. Tonight we're going to be entertained by Ian. Ian's been in at the ground level in genomics. He's worked in several of the major genome centers in WashU and then in England in the Sanger before he joined the Genome Center here nearly 10 years ago. He started out life as a molecular biologist, actually working on flies, those little fruit flies. And then he transitioned into bioinformatics, where he leads a number of bioinformatics efforts on campus about how to interpret the DNA sequence. As he'll tell you, we're faced by vast amounts of data, unprecedented amounts of interesting data. And the challenge is making sense of it all. But you're lucky, because Ian's going to tell you how.
Thank you, Richard. Yeah, I'm going to tell you a little bit about what DNA means today. It's a really interesting group of people that I have here today. To say, some people here who are professional scientists, other people here who I know from fencing. I don't know what you're doing here, Jerry, but it's totally cool. So let's just start off with what is DNA. I need to do some real background here to make sure that we're all on the same page. And usually you'll see something like this kind of structure here. And what is this exactly? You'll also see words like deoxyribonucleic acid. And what does any of that mean? Well, we're not really going to talk about the chemistry of DNA today. I'm going to talk about it in terms of a much more abstract concept. But I do want to give you a little bit of molecular biology just so you know what's going on here. So when people talk about the double helix, this is what they're talking about. That you can see this kind of helical structure, and there's two strands that are not symmetrically around each other. It really annoys me when people draw a helix. It's DNA helix, and they're symmetric, because when it's in water, it doesn't look like that. It has this kind of squashed look to it. Where this is called a minor groove, and this is the major groove over here. If you are a big Davis person, you might have heard of a band a while ago called the Minor Groove. That was really a great band. I just happened to be part of that. And Keith Bradnam here in the front, he was lead guitarist and singer of that band, the Minor Groove that was. And CDs will be sale after the show. So we talk about things having like a sugar phosphate backbone. That's what this part is here, what we're looking at here around there. If you think about DNA as a ladder, the outside parts of that is this sugar phosphate over again, and then the bases are the ladder rungs like that. And each one of these bases here has some kind of the difference in there. The different bases that go, and we're going to talk about those, are the different kinds of information that is in the DNA. This is the hereditary material for all of life as we know it. So everything on our planet uses this to communicate from one generation to the next. You'll also hear the term chromosome. Now, it's kind of confusing because you think about, DNA has these two strands, and then there's chromosomes that have sometimes pairs of chromosomes. Well, chromosomes don't have to come in pairs. If you're a bacterium, you have usually like one chromosome. It just so happens to be that if you're a diploid, like a human, you have pairs of chromosomes. So one of these chromosomes is going to come from your mom, another one is going to come from your dad. But if you were to zoom in on these little lines here, there is a double helix here. So this part is double helix, but you actually have two copies of this. One from your mom, one from your dad. And those copies are a little bit different from each other. They're not exactly the same. So we may call this, hey, this is chromosome 12 or something like that, and meaning that both of them are actually chromosome 12 in a way. Okay, one chromosome 12 from your mom, one from your dad. But each individual one of those, we can also think of that as a chromosome. If you were in a bacterial world, you wouldn't really have mom-dad kind of thing. So chromosomes can be linear or circular. We generally see them to be some kind of a linear thing because we're often thinking about ourselves. But probably most of the world has circular chromosomes. There's a lot of life out there, lots of bacteria and fungi, and there's lots of other things that maybe we don't know that much about, but the bacterial world outnumbers the non-bacterial world, and that's mostly circular DNA. But actually bacteria can also have linear DNA. They can have linear and circular DNA at the same time in the same case. So it's actually really interesting when you think about the bacterial world. It's not something we know much about. We hadn't historically. When you look in a microscope at a bacterium, it just looks like a speck. And the difference between one speck and another speck is very difficult to determine. Also, what we know about bacteria is generally what we can grow on a Petri plate. So a very fun experiment to do, which you'll see if you watch Mythbusters or if you go to your college microbiology class, is you'll take a Q-tip and you'll swab some surface and you'll spread it on some Petri plate and you'll see what grows on that Petri plate. And people have done these kinds of experiments many times, and then you end up being horrified at the kinds of things that grow on your kitchen sponge, and then surprised that nothing grows in your toilet. Part of that, so then if you could ask yourself, what would I rather lick, the kitchen sponge or the toilet? It's a difficult question to answer. One of the reasons it's difficult is because you don't want to lick the toilet, even if it looks like there's less bacteria. But that might be speaking to something that is somewhat unsaid in there, is that maybe not everything grows on a Petri plate. So how many of the things that if you were to swab and put them on a Petri plate, how many of those things that actually grow that were there? Because maybe things can live in one particular environment but not on a Petri plate. So probably something like 99% of life doesn't grow on a Petri plate. Maybe it's more than that. So almost nothing grows on a Petri plate. So when you do the experiment and you say, what's growing on my sponge, what's growing in my toilet, actually you're getting a very small sample of the things that could possibly be growing there. So the things that might be really bad for you could be on your toilet, or they could be in your sponge. It's kind of hard to know. And a microscope won't help you. But as I'm going to talk to you today, there is a modern microscope that can help you with that. Another term that you will hear is genome. Okay? Genome and DNA kind of go together quite. You can talk about, DNA, chromosomes. You're usually talking about all of the DNA of an organism. We call that, in all-encompassing DNA, its genome. Okay? So all the DNA of an individual is its genome. Okay, so when I talk about me and I'm saying, my genome, well, it turns out that every single one of my cells is very slightly different from every one of my other cells because of errors that occur in copying. But I still consider each one of my cells to have my genome in it. So it's kind of an abstract concept of the collection of DNA that's in a cell. So your genome is about, if you're human, which I think everybody here, 3 billion base pairs. It's kind of hard to think about how big is 3 billion when you think about 3 billion base pairs. Well, if you were to stretch that all out and it's a single thread, it would be about 3 meters long. A typical bacterial genome is actually much, much, much smaller. So we represent DNA as the letters A, C, G, and T. And we store DNA in text files, just like we could store books, Kindle stuff. Those things are in files. So then it has this analogy to the book of life. We talk about the genome as being the book of life, or the genome as being the book of life for a particular organism. Each one of them has its own book of life. And if you turned those pages of that book at about 1 per second, it would occupy you for about 3 weeks to do that. So here's an example of that. Let me put this on. So here is a file containing, this is actually sequence from the human genome. There are parts of your genome that may look 100% exactly like this. Okay? But in fact, every single person in this room is a little bit different from every other person in this room. About 1 out of every 1,000 of these letters is different between any two people. Okay? And if you were to page through this, 1 page at a time, every time I hit the space bar, we're seeing a whole new page of DNA. Like I was saying, it will. I hope you're patient. This is going to take me, to get to the end, it's going to take me 3 weeks. At 1 second each time. Yeah. So you get the idea of the scale of the human genome when you start to do this. It's like this literally, I'll get old before. Yeah. Which part is your favorite? I want to show you something interesting, though, about the human genome, and on many genomes, is that if you go, you'll notice that, I'm going to go up here, a whole bunch of DNA here has got letters, A, C, G, T, blah, 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 and then we get to a bunch of N's. What is an N? That's not an A, C, or G, or T. Normally when you're thinking about it, you go to school, you're taught that there's 4 nucleotides, A, C, G, T. N is meant to represent, I don't know. Okay? So it's sort of, well, I know there's sequence there in the chromosome, but I haven't figured out what it is. There's actually large parts of the human genome where we can't get the actual sequence out of it because it's repetitive, and it's hard to figure out how many repeats there are in those kinds of regions. So we have a good idea what's there, but we can't exactly say what it is. So when we put it in our file here, we usually, what they do is they'll say, oh, there's this big block of sequence, we'll just call it N for now, and then we can go back and do those kinds of harder parts later on. So there are many, many pages of N. You'll get bored after a while looking at all the N's, but then eventually, after you page through all the N's, you'll go back to stuff that looks quite a bit more like ordinary sequence. Another term that you will see a lot is gene. Genes contain the information to build specific parts of an organism. So it's not like people sometimes misuse the term and think, oh, there's a gene for the foot, and there's a gene for voting for this president or that president. No, well, that's not really the way genes work. Most of the genes that we think about code for proteins. And the proteins are the things that actually do stuff in a cell. So a cell, if you were to sort of microscopically go in and look at a cell, you would see all sorts of structures, things that look like long cables, things that look like scaffolds that are having actual structural roles. And then you would also see things that are motors, and you would see motors moving packages along these cables and changing the shapes of membranes and pumping different kinds of chemicals across barriers and kind of stuff. Those are all proteins. Almost all the work and all the structure that gets done in a cell is done by proteins. And proteins are these big, massive macromolecules made up of amino acids. But all of the information for how to make those proteins is encoded in the genes. It's in the DNA. I'll talk a little bit about how that's done. This is if thousands of years from now, when our society has broken down into something horrible and some new civilization comes up and they see cave paintings, this is the kind of cave painting they would see from my house. What are these diagrams meant to show? Well, they're meant to show genes. So genes have a direction. So this little arrow here is meant to indicate the direction. It's the promoter. So DNA, we were saying, it's a double helix, right? There's 2 strands. Well, you can actually read it in one direction or in the other direction. And genes tend to be on one strand or the other strand. But there are very rare cases when you can have the genes overlapping each other. But it's not common. It's sort of like taking a record and then doing it backwards and getting sort of satanic verses backwards. Most of the time, it's your imagination. But some of those times, it's real. So most bacterial genes look like this, okay? And there's a lot of genes in other, like us, that look like this, in which there is a single block of coding sequence. You can read it from one side to the other side and just interpret this and say, oh, I can understand what protein that's going to make. But most of the genes in your genome, probably on the order of 90 to 95% of them, something like that, actually have this structure where they're broken up into pieces. Okay? And so these pieces are called exons, the parts that code for things, and then the parts that don't have any kind of use are the introns. It's sort of like watching a TV, and the commercials are the introns. Or if you're reading a magazine, the advertisements are the introns. You sort of mentally take those things out and put the program back together, and that's what codes for the protein, these blue bits. But there's a lot of DNA, and a lot of people think that it's junk DNA. You'll hear that term sometimes, junk DNA. And a lot of times what people are referring to is the stuff out here that nobody knows what it does, or the stuff in introns that nobody knows what it does. I'm going to tell you a story later on about my research that shows that some of the things that we think is junk DNA is actually not junk. That some of these things in here are saying actually very important things to us. When we look at a schematic of a genome, we usually just draw a line. That's a genome, or a chromosome. Okay, it's just abstractly. And these are stick figures are our genes. But it's a representation, it's a very abstract representation for what it is. They are actually chemical structures in there, but we treat them and we look at them as just letters. And one of the more famous kind of discoveries that we had, I learned about this 27 years ago, and even then it wasn't cutting edge research. We've known about this for a long time, and that is the genetic code. There's actually more than one genetic code, but this is the common genetic code. And it tells you how to turn DNA into a protein. It's kind of amazing. It's read as triplets. So if the DNA has a sequence like this, you can translate that to protein by just saying, what does a C-A-T mean? You go to C-A-T. Oh, did I do this wrong? I did it wrong. It's leucine. I should have had the computer translate it. G-C-C, alanine. So that's alanine. T-T-C, T-T-C, phenylalanine, etc. So we can translate directly our DNA sequence into a protein sequence, and there's machinery in your cells that does exactly that. It reads the DNA, it looks at it in triplets, actually it goes through an RNA intermediate, but DNA gets turned into an RNA, and that RNA is read in triplets and then turned into a protein. Okay, so one of the most important languages of DNA is the genetic code. Now the genetic code through a lot of life is exactly the same. The genetic code from us to bacteria is largely the same. There are some obscure different organisms out there that have altered the genetic code a little bit here and there, but in large part it looks like this for almost all of life. This is sort of what a protein looks like. And if you looked at the atoms of it, it doesn't have this nice double helix shape. Every protein has a different look to it. This particular protein is used to phosphorylate sugar. So this is part of your, like when you eat something sugary, this protein complex acts on these tiny little molecules here. So every piece of sugar goes in there and gets modified, and that starts the glycolytic pathway. So proteins, these big machines, this is what is being made by the DNA. This is what the information codes for. These machines, and these machines do things like process all of these chemicals. Now sometimes you'll hear these terms. We use various kind of terms, genome, genomics. The 20th century is filled with all sorts of buzzwords that have these word "ome" in it, genomic. If we talk about all the proteins of a cell, we call that proteomics. So that's an ome that you'll hear about. And if we talk about all the small metabolic kind of things, all the small chemicals, there's room. Would you like to take a seat? Small molecules, then those are called metabolites, and we call this metabolomics. So some of our major omics are our genomics, our proteomics, our metabolomics. And just to plug the Genome Center here at UC Davis has pretty much a lot of the common omics, metabolomics, proteomics, transcriptomics. We have all sorts of technologies there, all in one building. UC Davis is very lucky to have a top-notch Genome Center. If you look at a microscope today, this is a modern microscope, you might use it to look at cells under it that might have some kind of look like this. The modern microscope of today does not look very much different from the microscope of 100 years ago. Largely the same thing. And it's for looking at very small things. But once you want to start looking at DNA, the DNA microscope looks a little bit different. When we want to start investigating what's happening with our hereditary material, we start needing lots of computers. So if you were to go to the Genome Center, I could give you a tour and I could show you our machine room, and it would look something like this. We call it Das Blinken Lights. There are lots and lots of computers, all blinking lights going along, generating an incredible amount of heat, using a lot of electricity because that's what's required to run the modern microscope of DNA microscope, is you need a lot of computing power. The sequencing machines that we have, that's how we actually look at investigate sequences by sequencing. One of the most important machines is this one. It's called the Illumina HiSeq. There's one down here that looks like it looks like Wall-E's date from Pixar movie. We have a machine here that does single molecule sequencing called the PacBio. This is actually quite large, be a good part of this front of this room. But we're getting into the point now where sequencers can be even something that you stick into your computer via USB. So the technology for sequencing is getting better and better all the time, and there doesn't seem to be any end in sight as to when we're going to stop making advances in this. And it's all great because we're being able to investigate things we never would have been able to see before. One of the things I was talking about just previously was bacteria. We can't study bacteria under a microscope because they all look the same. What we can do is we can sequence the bacteria and look at the bacteria as their different sequences. And that's something that could never have been done before. And now, with cheap computing and cheap high-throughput sequencing, we can do that. And that's an omic called metagenomics. So what do we get when we use these kinds of computers and sequencers and stuff like that? Well, this sort of looks like staring up at the night sky and seeing a constellation of little stars or something like that. And each one of these diagrams here represents a gene. We're just zoomed out so that you can't see very well that there's exons and introns. But the exons, I'll show you a close-up a little bit later. But what you see in this kind of display is lots of different genes. And the layout from left to right is one side of the chromosome to the other. Usually, but not always, chromosome is depicted horizontally. And there's two strands. There's a strand on the top and a strand on the bottom. What this one is showing is a lot of different interpretations, readings of that sequence to figure out where the genes are in there. It's not always clear. Here's taking a close-up. And we can see that here that this particular gene, the purple one, it starts over here on the left, ends over here on the right, has a fair number of exons and a fair number of introns. And there's another gene down here, which is going the opposite direction. The opposite direction genes are colored in a different color. But so there's a gene right next door to this other gene. The arrangement of genes on a chromosome is kind of interesting. Most of the times you'll have sort of genes on one strand or genes on the other strand. And every once in a while you have something really strange happen, like one gene can be on the inside of another gene. But it doesn't happen that often. But still, there's hundreds of examples in any genome. Unless you're a plant for some reason. Now when we start talking about DNA not as a chemical, but more and more as text and what we do with that. So for convenience, we symbolize nucleotides as letters, ACGT. They are not. They are actually chemicals. But inside a computer, instead of storing their entire molecular structure, we can just say A, C, G, T. There's only 4 of them, so 2 bits per character, if you want to think about it in terms of a computer. So how do we understand what the letters mean and the words mean? And so what I'm going to show you is the way we've looked at this in introns is that understanding DNA is a little bit like trying to understand a language. You don't really understand the language, but you can sort of interpret it. You'll see. So here's a quote from a very famous book, and anybody in here can read this. We all know the English language. It's easy the punctuation helps us discern the meaning. We can get the ends of sentences with capital letters and periods. This is not difficult. Does anybody know what book this is from? I always ask people because it's famous. It's Pride and Prejudice. Which everybody after, oh right, Pride and Prejudice, of course. That's like the first part of that, right? Because everybody's read it, of course. Now, all I've done is take out the punctuation. But this is very difficult to read, just because the punctuation is gone. Does anybody have an idea what this could be? This is Charles Darwin's Origin of Species. Also famous. Maybe not as famous. This is even harder. We don't have any punctuation. We've only got 4 letters, and we don't even know what the heck it's not even English. What can this possibly mean? It means something because the instructions are in here for building people and plants and worms and other kinds of things. All the information is there, if only we understood what any of it meant. So some of the problems with this is that the alphabet is so simple that one letter doesn't tell us anything. We have to take a collection of letters before we get to anywhere. There's no spacing, capitalization, punctuation of any kind. So where are we going to begin in our dissection of the patterns here? So I'm going to start by playing hangman. Okay. In the English language, we're about to play hangman. What letter should we start with? A is a good one. E is a good one. What do you think? By consensus? Who says A? By consensus, E. More people are saying E. Why E? Somebody tell me why E would be the best place to start with. It's common. It's the most common letter in the English language. That would be a good reason to start with that. So this is the frequencies of the letters in the English language. And E is, in fact, the most common. That would be a good choice. But in turn, and actually A is a good choice as well. T would be a good choice. There's a number of good choices, but in fact, E is the best one. So we don't need to know that much about English to be able to play hangman with some level of accuracy. Look at English and Spanish. They are really, really similar in terms of their letter frequencies. Well, there's more J's in Spanish than English, and more K's in English than Spanish. But overall, there's the same kind of pattern of usage of letters in English and Spanish. They're not that different from each other. But I have no idea what this says. I just pulled this out of something, some text, probably some free text from the Gutenberg project. But I have no idea what this means, because the letter frequencies don't help me anything when it comes to understanding words. The meaning is in the combinations of letters to make words. It's not just the letters. You can't know the letters and the frequencies of them and suddenly understand Spanish. Words are the things that distinguish the languages, and not the letters. And that's the same kind of thing we're going to talk about when we go into talking about the patterns that exist in DNA. We're going to start looking for patterns that exist in words and not into letters. But is this even true in Elvish? That there would be... What am I talking about, Elvish? What's Elvish? Elvish is a made-up language. It's not even a real language. So J.R.R. Tolkien made up a language of Elvish, and he's written books on that. And I don't think anybody here probably knows what this says. But it says... This is where I had that posted. Something like, I think that is... go with the stars or something. So this is where I want to break out just a little bit and actually play with the computer a little bit and show you something fun. So if you're J.R.R. Tolkien and you want to come up with a new language like Elvish, you are probably a linguist and you study languages and you understand like the kinds of things that you're trying to get out of a language. If you're not, if you're a computer nerd like me and you want to come up with, say, words that would work in the let's take a look at how we can make our own Elvish up. So one thing I might do is I might start by downloading text from different languages. Where can I do that? I can do that from the Gutenberg project. Free books. You can get all sorts of books for free at the Gutenberg project. And you can read them. For example, this is Jane Austen's Emma. So this is where I yanked that thing out of earlier. Yes, no problem. You can imagine it'd be laborious, but you could figure out how often every letter occurs in the English language using any of these books, and probably, regardless of the book, you would probably get a very similar frequency of letters in those languages. And that would also be true of any of these other languages. But if I were if I want to generate new Elvish words, can I just use the frequencies of the letters? Well, that's a good place to start when you're playing hangman. But let's say we're playing hangman and there's two letters in a row, E and E. Okay? We know that there's not going to be, well, because of the rules of hangman, there's not going to be E's on either side of that because there's no language, there's no letters there's no words that have three E's in them. There's certain rules we have. If we have a Q, well, we know that the following letter is probably a U, okay? And the preceding letter is definitely not a Q. Okay, so there are certain rules that we have in languages that we don't just use the letters, the letter frequencies when we're playing hangman. We also think about the other types of rules that we know of language. Can we observe these same kinds of rules when we look at other languages? So let's take a look at Finnish. This is the Finnish language. I have no idea what this means, but it looks Elvish to me. And apparently it did to J.R.R. Tolkien, because he actually made his, it's actually a combination of Welsh and Finnish is where he got his ideas from. We have to get past this header material. And then we get into this kind of stuff, which is Welsh, which apparently to Tolkien looked a bit Elvish. To him, it was exotic. So he mixed those two things. So we can actually do the same thing. I can make my own kind of program that does those kinds of things. What this program is going to do is it's going to take any of these particular files, look at the frequencies of the letters, and then build new words with the exact same frequencies. But the important point is, it's not just using one letter at a time. The probability that it finds that it's going to put a letter in is dependent on the letters that are around it. So for example, in German, if the letters have lots of Z's in them or F's or something, and they're next to other things that have Z's or F's, that's what it'll look like. So if we do this first, let's say we first do this list English, and we do this with Emma, and we tell it to generate a whole bunch of words. Some of these words actually are English words. It did this randomly, but came up with some things that are real words. But other ones, explos is not a word. But based on the rules of probability, it wouldn't be a bad word. That's actually, if you were coming up with new words in the English language, this taking apparently is not a bad one. Now, we're very critical of ourselves when we do this because we know the language too well. But if we wanted to do this with another language, for example, Principia Mathematica, very famous. Let's do this with Latin. There's some great new Latin words. As far as I know, obviously this didn't work very well. It's some kind of number, but we have to do some post-processing on some of these. But some of these words, certainly, that's Latin for ham sandwich or something. That's clearly mother or something. Anyhow, so we can play the exact same words, these kinds of games with any language we want. We can even do it with, let's say, put both the Welsh and the Finnish ones in there. And now these are a fusion of Welsh and Finnish probabilities. And so these words, like whatever this is, would be a legitimate sort of, if we're coming up with, if we're writing a novel and J.R.R. Tolkien kind of way of doing it, and we needed to come up with new words, as a friend of mine has done, as it turns out, and he came out, he needed words for his book, and this is exactly what he did. He generated them by looking at the frequencies of known languages that looked Elvish to him, and then did that. So if you thought, for example, another language, for example, what else do I have in here? I have Old English. So clearly, Old English plus, do I have German? I don't have, German. If we put those together, that's Orcish. Those are Orcish words, clearly. Well, to me anyway. And if I was writing my book, I could get away with that. So this game that we've been playing here is that we don't really, the whole idea is that the frequencies of which pairs of letters come together is something that's important, not single letters. So what are the words of DNA? We don't have any punctuation in DNA, so what can we do to break up the words? Well, we assume actually what happens instead is that we're not going to use any punctuation. Every possible combination of letters will make a word. We just have to choose what size word we want. So we can say, let's choose a word size of 5. And in the language of the computer nerds, we'll say, k equals 5, and we'll call it a Kmer. Okay, so when k equals 5, what are the various words? The first word is TTGCG. That's just these first 5 letters right here. And if we move over one, we get the next word, and the next word, and the next word. So we scan along, sometimes we call this a sliding window, along the DNA, 5 letters at a time, and just pull those things out. And those are all the words that we can find in the DNA. Now, by that self, that might not be very interesting, but it's the frequency with which those words occur that is interesting. So if we were to look at English and say, what is the most common word in the English language? And we know that space is separated, it's the. But if we do the same kind of strategy, but don't know where this, and just take all the spaces out, it still turns out to be the. But the most common 4-letter word is T-I-O-N. It's the end of whatever, speciation or something. So this is an analysis of the origin of species, and you end up as the most common 7-letter word as being species. Maybe not too surprising if the book is the origin of species. But we've learned that without knowing any punctuation at all. All the commas, everything was removed from that, all the spacing, and yet the most common thing that came out, it was species. So we've done something incredibly crude here, but yet those patterns are emerging. So here's our overall procedure. Can we determine the important words encoded in DNA, and what do the words mean? So let me tell you now about some research that we've done at UC Davis. It's mostly between my lab and Alan Rose's lab. Alan Rose does a lot of molecular biology. He works on this plant here, Arabidopsis thaliana, and I work mostly on the computer side. So in biology, a lot of biotechnology applications, people want to take a gene from one organism and stick it in another organism. Maybe it confers some kind of disease resistance or something like that. So for example, maybe you work with a worm, and there's a gene that you like from that one. Generally, you can't take that whole gene and stick it from one organism into the next, because the introns can be quite large and it can be too big to transport from one place to another. And so what people do is they take the intermediate of this. This actually comes from the messenger RNA, which has all those introns removed from it, and that's called the cDNA. And they take that and they put it in what they call a vector, and they take that from one organism to another organism. So you take the gene out of this worm and you get it into this plant. And this might confer, something like frost resistance or whatever, some beneficial kind of thing. Or maybe you're really excited about providing nutrition for people, and it's you're trying to do something like with golden rice, for example, where you're trying to cure night blindness. But there's a lot of reasons people might want to move one gene from one organism to the next. So in its new context here, we put this in this other organism. Now the gene is in there. But what we found, and many people have found this over and over, is that this gene, all by itself, the expression was something low. We'll call it 1x. It's very low. It doesn't really work. So when you've made your golden rice, it's not very golden. It doesn't make enough carotene for anybody to care about. They need more of it. But when you put an intron in there, for some reason, and some introns, you get a whole lot more expression. And this has been a puzzle for a very, very long time. So one of the first questions is are all introns equal? Can we put any kind of thing in there? And the answer to that is shown in this typical sort of old school molecular biology, in which we're looking at each pair of these is a different construct. So this one over here, no intron, it's just that short block. Then we put a different intron in, and then another intron. Different introns are going in there. And you can see that this blackness is showing that there's a big difference. We're actually looking at the accumulation of RNA. And the intron makes a huge difference. This intron. The other intron doesn't really do anything at all. Okay, so not every intron can do this. Some introns are important, some introns are not in that way. So one of the main questions is, what is different about this UBQ10 and Korf15A introns? The UBQ10 one is incredibly highly expressing. Korf15A doesn't do anything more than no intron. Are there language differences? Can we find words that are in one intron that are not in the other intron? There's one of them speaking Elvish and the other one is speaking Orcish. What subsequences are necessary for enhancement? Is there some part of that? Can we dissect in some way and figure out what it is? And what subsequences are sufficient, meaning that we could pull out and put somewhere else and they would also work? So we can do molecular biology experiments like this. We'll also get to the bioinformatics kind of stuff. But in molecular biology, if you're doing genetics, when you're trying to figure out if something works, you try and break it first. So if you hit it with a hammer and it no longer works, and you say, I know what it does now. This doesn't make them very good mechanics necessarily for your car, but they can very easily determine the essential parts and non-essential parts of your car. So if this is the intron, remember this is that piece of junk DNA that we stick inside this construct, and that we're generally getting a lot of expression out of, the idea is, with these deletions, is to remove part of the sequence, to break it, to hit it with a hammer, and see if it no longer works anymore. And the strange thing is in all these cases, now we're looking at an assay, an enzyme assay, where the blue color means, yay, lots of expression, and the white color means very little expression. And what you can see here is that when you delete any part of this, it still works. Now, if you imagine that the intron is a sentence, or a paragraph, or a book, or something like that, you would probably think that you would lose a lot of the meaning by taking out 1/4 of the book. You might not even be able to figure out what happens anymore. Here, somebody just removed the end of the book. You'd be frustrated for a very long time. But in this case, it doesn't appear to matter. All parts of the book tell the same kind of story. Turn this gene on. So it's a bit weird in that way, because most of the time that doesn't happen. And if this was a protein, it would definitely no longer work. Another experiment we did is to say, let's take this intron that doesn't work, we already know it doesn't work, and put in pieces from this highly enhancing intron and stick it in there and see if can we boost up gene expression just with any part of this. Can any part of this tell us some part of the story? And the answer is yes, all of these boost expression, some of them not as much as other, but they all improve the expression. So meaning that whatever these signals are, they're sort of all over the place. It's not what we expected. So probably the most critical experiment that was done was that he took that intron, that piece of junk DNA, and he moved it down the transcript. He moved it into different positions. You would think, well, why would anybody do that? I would think, why would anybody do that? But that was actually the critical experiment, because as you move that farther down, we're looking at this kind of dark smudge here, that smudge starts to go away. The intron has to be near the beginning, the promoter, that little arrow, square arrow that I drew on our anatomy of a gene. The intron has to be near that, otherwise there's no effect at all. So it was a big puzzle for us. So some of the properties of this is called IME, intron-mediated enhancement. I didn't tell you what that was, but not all introns can enhance gene expression. Whatever the signals are, whatever those words are, they appear to be all over. They're redundant and dispersed. The introns have to be near the promoter to have an effect. Splicing is not required. That's the part of removing stuff, which we don't really need to talk about. And no increase in transcription initiation, meaning that the promoter at the beginning is not in any way really affected. So we came up with a model for how this works. And some of this is going to be sort of mumbo-jumbo molecular biology speak. But the idea is that in the case of whether you have an intron or not, everything starts exactly the same. You have a promoter and it starts moving. I mean, the promoter starts the polymerase going, the polymerase starts going down the gene and starts to make a transcript. That in the presence of an intron, it keeps going, and if it doesn't see an intron, it tends to fall off. And by falling off, it doesn't complete it. And so when you have an intron in there, the whole transcript gets made. Your book gets published, and if you don't have an intron in there, it doesn't happen. It doesn't go out there into the rest of the cell and doesn't make a protein. So this one, when you have an intron in it, it can make a protein, and this one it doesn't. And it's dependent on whatever, some kind of word, something different about that sequence of the intron. So how do we find these differences? So we're going to look for differences in enhancing and non-enhancing introns. Our data set is really small, so we're going to use the entire genome is our idea. And let me give you the idea here is that, let me go back to show you something here. So remember I said that when you move the intron farther down, it no longer has an effect? So the intron near the beginning of the gene, this one, may be different from all the other ones. The one up here is near the promoter. That's the only one that should have any kind of enhancing ability. So the question is the intron here different from the other ones. And the technique we're going to use to do that is the exact same kind of technique we were using when we were thinking about how the different languages work. So, for example, is the intron that's near the promoter speaking Elvish and the one down at the end speaking English? How can we determine that? By the frequencies of the words. So what we're going to do is we're going to count all of the words, all the k-mers in the introns near the promoter, and count all of the k-mers in the things that are farther away, and just look, what is the frequency at which you see those different kinds of words? It could be the same kinds of, like, what is the frequency of people who are older? Like, let's say older people are at the beginning and younger people are at the end. The word frequency over here, older people, you know, we're smarter. We don't use like and such. But the younger people, they're just filled with like. I'm an older guy, so I can say that. I'm not that old, I know. Just trying to make a connection with our older crowd. There's not too many young people here. And they're young anyhow, they can take it. Okay, so this is what we're looking for, is these word differences. So let me show you what I've got here, which is that this word, CG, okay, is 2 times more likely to occur near the beginning than the end. Okay? And this word down here, GCACC, is 1.25 times more likely to occur down there than up here. But these are actually log odd scores, so it's actually 2 to that power. But anyhow, it's 2 to the 1.9, so it's more like 4, and this is more like 2 times. But so what this means is that there's some words that are spoken more frequently in here and other words that are spoken more frequently down there. They're speaking different things. Not every intron is the same as every other intron. There are actually different dialects being spoken here. And, but if you were to compare all of these ones to each other, they're all pretty much the same. It's just the first one that appears to be really different. So we came up with this very simple equation. But there's only a couple of parameters. These ones you don't have to worry about. It's just the saying that I'm going to explain this to you, and you're all going to walk away understanding this, okay? And that'll be the biggest triumph of my teaching career, okay? An intron is going to be some sequence, some body of sequence left to right. And we're going to count up all the various words of there, okay? So what we're going to do is you give me any sequence, and I can tell you whether that thing looks like it came from English or Elvish, okay? So let's say P over here. This is the probability of a word being Elvish, and the probability of a word being English is Q. So Elvish over English. If I come across a bunch of random text, and I see the word tenuvial, is that English or Elvish? Well, okay, I'm just going to say that's Elvish. So score 1 for Elvish. Then we go along and we see another word, and it's Adam. Well, Adam sounds English. Score 1 for that. We do this one. So we just keep adding up the words as we go. Of course, they're not words that are separated by spaces. They're actually words that are like GCATTC, GCCATT, whatever. We're going to go along there, and we're just going to count up how often they occur in the frequency which we expect them to occur in one language versus the other. And then when we take the logarithm of that, then it becomes like, if they're exactly the same, then the logarithm of 1 is going to be 0, so we balance. But if this will have a positive score, will mean it's more like this, and a negative score more like this. So if we have a positive score, it looks more like Elvish. If we have a negative score, it looks more like English. And if Elvish was, for example, our enhancing state, we're looking for positive scores there. So any sequence because that you give me, I can score in this way because I know what the two languages look like. If I have a body of text that looks like, hey, I've got Elvish text, I've got English text, I can figure out what the word frequencies are. That's what the P and the Q are. Just how often do those words occur in each one of those languages. That's just a simple matter of counting them up and dividing by the total. And I do the exact same thing right back here. I counted up all the words that occurred in the introns that enhance and all the words that introns that didn't enhance. I put them into a big table, and I got for those the log odds of those. That's essentially what is right here. And so I am just going to count up all of the words and say, did they come from one place or another? And so it's like, oh, you know what? We've got 5 words from Elvish and 2 words from English. More Elvish than English. That's all that this is. It's just saying which one of those things does it look more like. Okay? It's just a mathematical formalism for saying, you know, let's just count these up and which one, where's the balance lie? If we plot this out on a graph, okay, and we have the score here, the I-meter score, more positive means it looks more like it came from introns at the beginning, okay, and we plot this versus how much expression do we get, which would be how blue are those Petri plates, or how black were those bands on that other assay, the autoradiograph, then we get a pretty straight line, which says that we're able to predict. If you give me a sequence, and I don't know anything about it, I can tell you how much it enhances. I have learned to speak intron. Okay, you can give me anything and I can tell you whether it's going to work or not. It's kind of amazing because I have no idea what the actual mechanism is. I'm just using computers, a little bit of computers, a little bit of programs, and I can figure this kind of stuff out. I can show you the program for this. It's ridiculously simple. It fits in 100 lines of code. So we took all of the introns and we plotted them out. So the position, how far away from the promoter it is, and then what the score of that intron was. Okay? And you can see these ones that are red up here, we've identified those ones as we saying they're high scoring. Most of those are near the beginning of genes, as we expect. But not every intron enhances gene expression that's near the front. Some of them more than others. Okay? So what we did is then we took some of these ones that are red ones and we said, what kind of pattern do we find in that red one that's interesting? Like, could we find that species comes out of the origin of species? What common pattern comes out of there? And this is the pattern that comes out. So this is an interesting display that shows the frequencies of the letters. The higher the letter is, the more common that letter is in that particular position. So a sequence that is GAT, CTG, plus some other stuff on the other side, would be something that would be expected to enhance gene expression. Somehow this is improving the expression. But there's variants of that. So sometimes you can put a C in here, and each position is a little bit variable. But this is one of the ways that we display uncertainty when we're saying that this word and other things similar to it are responsible for some kind of effect. We use this kind of motif kind of pattern. It's not exactly equal to the frequency at this particular position. If they were, all of them would add up to the top bar. It's actually the information content at each point, which we could discuss if we had 20 minutes, but I would need a chalkboard to do it. So what we can do is we can then say, what if we were to take that intron, Core 15A was the one, didn't enhance expression at all, and edit it, change it by putting that motif, that thing that I just showed you here. We're going to take this sequence, things like that, and put it all through this sequence here that does not enhance expression at all, and see if it does. So here's the original sequence. Here it is modified. We've turned this thing that was not really enhancing expression at all into something that's very powerful. And in fact, Alan has done this and embedded even more of these in here, and has made now an intron that is probably the most powerful enhancing intron that is known in any organism. And you can imagine that this could be very good for people, for example, who are making golden rice, because it would make their golden rice that much more golden. It would have much more nutritional value, or maybe you wouldn't have to eat quite as much of it. Not that anybody is actually eating golden rice. That's another story. There's all sorts of legal problems with that. So I want to just summarize here that in this story I've been telling you that proximal and distal introns are functionally distinct. That's pretty much how we started this story. And it's like saying, you know, English and French are functionally distinct. They are different things, as are the introns here and the introns here. If they were not different, we wouldn't be able to do any of this. Some words preferentially occur in proximal introns. There are certain things like that pattern of G, A, T, C, whatever. Those things happen in one place that don't happen as much in the other place. We can make these motifs, these patterns from these high-scoring introns, and then turn non-enhancing introns, things that are, you know, basically we've got a book and then it's in English and translated into something Elvish. It's not a very good analogy. But we're able to turn it's really kind of junk into something that's quite golden. So we learned to speak a little bit of this intron language because we had a whole genome to work with, the Arabidopsis genome. We used a tiny bit of math, just that one equation, and a little bit of programming to show the program. It's actually quite small, and a little bit of computer time as well. And it was done actually even on something as small as a laptop. It doesn't require the big giant hardware or anything like that. This was just one of the languages that I was showing you. I was showing you the language of introns in plants. But actually, there's many, many, many different languages that are present. Aside from the coding sequence, there's the promoters, what gets genes started. There's in every genome, they have different ways of saying the same thing. So even in one genome, way it specifies protein is slightly different from another. They have just sort of different dialects that way. So there's actually much more to do in deciphering the genome. We're really at the beginning of this. We have the capacity to sequence genomes very cheaply now, but we have no idea really what any of this stuff means. And it's amazing in some ways that using this crude bit of math and a little bit of computers and stuff like that, we're able to decipher some of what's actually there. But the real goal of understanding what the various base pairs do is a goal that we won't be able to achieve that for decades. We're many, many years away from being able to figure out what it all means. It's very complicated, but it's actually a great playground for people who are interested in this kind of stuff. It's a great time to be a molecular biologist and a bioinformaticist and that kind of stuff. Some people who funded this included NHGRI, Human Genome Research Institute, the USDA, Alan Rose, a close collaborator here at UC Davis, Keith Bradnam here in the front row or second row. He did a lot of the actual work, and some other people, students and postdocs and other people who've worked on this project from time to time. And I will take whatever questions you have at that now.
Can you tell us a bit more about the promoters?
Promoters?
What are they and how do you determine?
Promoters are what sort of start the ball rolling. You won't get a protein made unless the promoter is somehow active and says, oh, it's time to start making this particular protein. The patterns that exist at promoters tend to be clusters of transcription factor binding sites. So a transcription factor is a protein that binds to the DNA and says, let's get going. And usually, it's not enough just to have one. You have to have a cluster of these things. You sort of have to get enough people together, quorum, saying, hey, let's make some changes around here. And not just one person will do it. You need several people around there to do it. And it might take an interaction between more than one person. It's like it might have to require both political parties to actually say that we need to do some work on Highway 80. And so both of those kinds of signals will occur up there. Unless both of those proteins are present, you won't get it to be made. And this is the reason why you have, say, a liver cell is different from a muscle cell, is because inside those cells you will have different transcription factors, and the ones in the muscle cell and the ones in the liver cell will be telling different genes to go on and off. So most of the control of what happens in a cell, the various different proteins that are there, is going to be set up by what's happening at the promoters. So the promoters are actually really a critical part of when genes go on and when they go off and how much of the, that's the sort of main brain of everything, of all the control.
Introns have been labeled junk, but
Yes.
are they not junk, or are they just something we don't understand?
Well, I think most of the stuff that's junk is stuff that we don't understand, and some of it's very subtle, has very subtle effects. So it's not clear exactly what it does, or we can't put a finger on it and say, I know exactly what this does, but it probably does have some function. Otherwise, you wouldn't keep around a lot of this junk. But certainly there are cases where you could probably say that some parts are more junky than other parts. Definitely the stuff that's in introns, a lot of times you can remove large parts of it and it won't matter. So it's not playing that important a role. But there are probably some subtle effects for most of the sequences in there. There's a lot of news about, how much percent, what part of the human genome or any other genome is junk versus functional and stuff like that. And it's a very difficult number to come up with. Somebody came up with this number of like 80% or 85% is functional. The number is surely wrong. Depends on what you mean by functional. You can't just label something as functional or not functional. It's very difficult to do that. But a very small part of it recognizably does something very important. Most of it does not. Yeah, and some of the stuff that we're doing here, we're finding some of the stuff that's junk is, but the thing is, like with that intron, we could remove different parts and it still work. So on that way, you might have just interpreted that it's all junk because I can remove different parts of it and it still works. But on the other hand, it was probably pretty important. If you remove the whole thing, it doesn't work at all. In the model, like why does it stop? So the model is that once the polymerase gets on and starts moving, that there's just a random probability that it falls off, kind of like a half-life, that it's going to fall off. And what happens is when it hits an intron, then there's something in the intron, perhaps interacting with those signals, that then tells the polymerase, maybe phosphorylates the polymerase or something like that, and says keep going, and then it doesn't stop. It goes from this kind of wishy-washy thing into a very, sort of processive enzyme. So that's the model that we have for that. There's some kind of communication that goes on between something that's in the intron, something's grabbing onto the intron and telling things. That's the model.